Okay, so our second speaker is Professor Gordan Lauc from the University of Zagreb here in Croatia. So Professor Lauc has numerous achievements and responsibilities, including being the director of the National Center for Excellence in Personalized Healthcare, and he's also the CEO at the Genos Glycoscience Research Laboratory. And he will be talking about glycans as biomarkers and functional effectors in cardiometabolic diseases.
So thank you for the introduction. And in the next 20-25 minutes, do we have a timer? Oh, it's only I see there. Okay. In the next 25 minutes, I will tell you something about the glycans and what we have done to develop them into biomarkers and functional effectors of cardiometabolic diseases. Not so many people work with glycans, so I will make a short introduction about what are glycans. So in the middle, we see revolving the most famous molecule at the planet at the moment, the S-glycoprotein of the SARS-CoV-2 virus, and everything hairy on its surface are the glycans. So let me just try the pointer. So these are glycans on the surface of a protein. Actually, the protein on its surface is more glycan than a polypeptide. And unfortunately, many scientists still look only at the polypeptide part. This is the figure on the left. And the best analogy I can make is this poor bird without a single feather. So you can plan an experiment well, you can execute it perfectly, you can study the eyesight of that bird, you can study even the bird walking, physiology of the bird, but you will never see that bird flying, because bird without its feathers cannot fly. The same goes for proteins. Protein without its glycans does not function normally. And if you look only at the polypeptide part, you are missing a big part of biology. And it's not only the S-glycoprotein of the SARS-CoV-2 virus, it's the majority of proteins that are glycosylated. But one word of caution here, glycation is something completely different. Glycation is a chemical reaction between the glucose and amino groups on the proteins. We measure it as a glycated hemoglobin. It's a part of a diabetes diagnosis, but the glycation is a random damage. It's taking a shotgun and firing at the protein. Glycosylation is something completely different. Glycosylation is a sophisticated biological process which enriches the protein structure and the protein function. And if you want to understand biology, we have to look into glycans. The problem with glycans is that they're difficult to analyze. Complex chemical structures require many chemical steps to analyze them, especially in high throughput. And we were first who went in that direction. We published our first large throughput, high throughput glycomic studies over a decade ago. And if you want to see our final progress in that field, there is a poster, 43, I believe, that you can visit in our poster session. In the last decade, we analyzed over 150,000 glycans. Actually, there is a review coming out in the Chemical Reviews in a few months, which listed 190,000 glycans which were published as a part of high-throughput glycomic studies. Over 160,000 were generated in my lab. And we try to do it in collaboration with the leading researchers all around the world so that we do not waste our precious analytical capacity on samples which do not have adequate clinical or other omic data. And one of the proteins we are focusing on is immunoglobulin G. Immunoglobulin G is practically regulated by glycans. Not the binding, not the FAB part, but the FC part. What will be the effector functions, to which receptors will IgG bind and which branch of the immune system will it activate is by large determined by glycans. So depending on which glycan structure is attached, we will have activation of different receptors. And the field of immunology finally started to appreciate importance of glycosylation. And for example, our review, which was published in 2018, is still the most downloaded and the second most cited paper in cellular immunology. Important thing about IgG glycosylation is that its regulation is very intricate, and it involves over 40 genes. Around 25 of them have been published, the others are our unpublished data. We published a series of GWAS papers mapping the genes which work together to decide which glycans will be put to the IgG, meaning will IgG be pro-inflammatory, anti-inflammatory, will it activate ADCC or some other aspects of the immune system. And important thing to understand here is that putting a different glycan on a given glycosylation site has functional consequences which are analogous to coding mutation. We know that coding mutation can kill a person. Having a wrong letter in our genome makes the protein different, and the protein may be completely non-functional. The same is for alternative glycosylation. So putting the wrong glycan on a given glycosylation site can make protein completely non-functional. But this is not encoded in a single letter in a genome. It is encoded in a network of dozens of hundreds of genes, and all their polymorphisms which integrate into the decision how to glycosylate the protein. And actually, nearly a decade ago, Vladka and I speculated that glycans are the third revolution in evolution. And subsequently, we collected a significant amount of data suggesting that this speculation was actually correct. So we looked at a collaborative cohort of mice generated by Grant Morahan. So he started with 8 very different founder strains of mice, crossed them for 3 generations, and then inbred these individuals into strains. So he generated the strains of cousins. And some of these strains were completely different than the rest of the strains, based on IgG glycan composition, meaning the structure and function of IgG, which was achieved without any mutations, just by reshuffling the allelic variants, generating the novel networks which regulate IgG glycosylation, and in this way generating novel chemical structures which are being inherited as a complex trait. And it's not only allelic variants, it's also epigenetic variation of the same genes, which can have very similar effects, and also direct environmental factors which are affecting the glycome. And when we looked for a pleiotropy, we saw very extensive pleiotropy for the genes which regulate IgG glycosylation with multiple different diseases. So for IgG and the first, I think, 26 genes which we identified in the Science Advances papers a couple of years ago, we have 94 different traits, different conditions and diseases where we have this pleiotropy. Some of them are expected, like lupus or type 1 diabetes, but some are completely unexpected, like a cholesterol concentration or some forms of dementia. For the latest update, there is a full poster IP56, I believe. IPS6, sorry. And interestingly, these regulatory networks are protein-specific. So we did a parallel GWAS of transferrin and IgG, which have very similar glycan structures. So chemically, the structures which are produced by the glycosylation machinery are nearly the same, but the regulatory networks which decide which structure will be added either to transferrin or IgG are completely different. This was published in Nature Communication, I believe last year, actually earlier this year. And what we are now trying to do, we are trying to understand how is this actually happening. And here we are collaborating with Vladka Zoldos and her team. They developed a versatile CRISPR-Cas tool which can be used to activate, deactivate gene, methylate, demethylate specific region, introduce different histone modifications, and they can actually even have 2 opposing modifications in the same cell line, so we can really play with the genes, activate and deactivate them, and see what actually happens. There are a couple of posters which you can visit coming from her group, and what they developed for us is 2 cell lines which were stably transfected either with activating or deactivating CRISPR tools with plasmid, which is containing the genes for IgG, having a light chain. So when we transfect the HEC3 cell line, we are putting in the gene and the specific guidance RNAs, which then either activate or deactivate a given gene. And then we looked at IgG glycosylation to see what is happening with IgG glycosylation in this in vitro system when you modulate specific genes. And this seems to be a very popular tool among the people who were judging the posters here, because Carlo got the Young Investigator Award. He had his presentation yesterday, so if you missed it, you missed it. But he's very motivated to talk about it, so he actually painted his color blue, so you can recognize him. So guy with the blue hair, he's the one who did that. And there will be another short presentation by his mentor, Professor Vojta, on Monday. So there is a chance to hear more about it. And one example we have already published, we have used these tools to look at the downstream signaling pathway between estrogen and IgG glycosylation. We know estrogen is a very strong effector. I will talk about it a little bit later. And here we use these tools to map the downstream genes to see which of them have which effect on IgG glycosylation in vitro system. I emphasize the in vitro system because many of the genes which we know that regulate IgG glycosylation are actually not the B-cell genes. They're working in other parts of the immune system, but the consequence is change in IgG glycosylation and alteration of IgG function. From the epidemiological perspective, first things we learned when we started to look at the large cohorts is that glycans change a lot with age. And first, this was a problem for us because we wanted to look at the diseases, and so the patients and controls had to be perfectly age-matched, which is not always easy. But then we realized there is a huge potential in knowing that IgG glycans change with age, and then we developed a glycan clock of aging, because what we learned is that the age we predict based on glycans is actually revealing a lot about our lifestyle, because people who are aging faster usually have all the biomarkers of unhealthy lifestyle, while people who are aging slowly based on glycans would have a healthier lifestyle. And here we even developed a commercial product. Of course, I am heavily conflicted here, so you have to be cautious in judging what I'm saying. But what we know that younger people have glycans which are suppressing inflammation, and the older people have glycans which are promoting inflammation. And the pace of changes from suppressing to promoting inflammation is by large defined by our lifestyle. If you're interested, Glycan Age is one of the sponsors of the conference. They have a booth just outside of this room. When we looked all around the world, and we looked at 27 different populations all around the world, we saw that people living in different parts of the world are different. And the strongest predictor or the strongest correlation was with the expected lifespan. So people living in poor countries where the expected lifespan is shorter would change from young to old glycans much faster than people who would live in countries with a longer expected lifespan. And the other very strong effectors which affected IgG glycosylation were different diseases. But different diseases. We really studied dozens of diseases, and when we look at the glycans which are part of this Glycan Age index, and this is galactosylation and sialylation, we see that in the majority of diseases, and here we have things like IBD, RA, lupus, colorectal cancer, diabetes, many different diseases, HIV, they change in the same direction as aging. So people with different diseases have glycans which look older. And the change in glycans often comes up to a decade before the disease is diagnosed. So what we believe here, we are not looking at a disease-specific biomarker, but we are looking at the molecular process, which is regulation of low-grade chronic inflammation, which is actually contributing to disease development. So with our lifestyle, we change our glycans to become more pro-inflammatory. This pro-inflammatory process, this low-grade chronic inflammation, is putting us under additional stress, and then depending on the genetics, environment, epigenetics, we will end up in one of the different diseases. And since we knew that expected lifespan is something which highly correlates, then we first suspected that the cardiovascular disease is something which is interesting to look at, because the cardiovascular diseases are still the major killer. And when we looked at the first two cohorts in England and Scotland, we saw that glycans today highly correlate with the cardiovascular disease risk score, meaning the risk of getting cardiovascular disease in the future. And we published this in Circulation Research 4 years ago. And using that data, we went to the EPIC cohort. An EPIC cohort is a fantastic cohort collected in Europe nearly 30 years ago. They collected 300,000 people with a clinical follow-up. We got samples from the German part of the cohort. And in German part of the cohort, out of 27,000 collected samples, around 460, 70 of them got either heart attack or stroke. So we had a collected prospective study of samples at the beginning and then a clinical follow-up for a decade to see who will get a heart attack or stroke. And IgG glycan composition, just glycans attached to IgG, were as predictive as the entire AHA score. And in women, even a little bit more predictive than the entire AHA score. And we saw a similar thing for diabetes. In the same cohort, we saw that the glycans, but not IgG glycans, the total plasma glycans, were also as predictive as the entire German diabetes risk score to predict diabetes. So the composition of a glycan today predicts whether we will develop heart attack, stroke, diabetes up to a decade in the future. And we believe this is by large causative, because there is a lot of evidence suggesting that changes in glycans may actually contribute to disease. So this is work by others, not by us, where it was identified that old glycans, the glycans without sialic acid, are actually blocking the release of nitric oxide from the endothelial cells, which is being promoted by the young IgG glycans, which contain sialic acid. When we looked in our population cohorts, and these were 2,000 twins tracked for 20 years with the 3 sampling on average every 7 years, we saw that these changes in the glycom come before the hypertension starts to develop. So we see first the changes in the glycom, then we see hypertension developing. And the most interestingly, but for now this is only the mouse study, we can prevent hypertension by improving IgG glycosylation. So if you put mice on a high-fat diet, they become obese, they lose IgG cellulation, so their glycom becomes old, and this promotes hypertension. If you feed mice with a MANAC, N-acetylmanosamine, which is promoting cellulation, the IgG does not lose cellulation. Mice, of course, become obese, but they do not develop hypertension. This was published in Circulation 3 years ago. Also for diabetes, there is a lot of evidence that glycans regulate glucose sensing, glucose uptake, insulin secretion, insulin sensitivity. So we looked in our population cohorts, and we saw that glycans have a significant predictive potential to predict diabetes in the future, but not in a kind of disease risk condition, altered glycans, but as a gradual change from health to disease. So what we see here, we see red dot is the glycans in people who did have either diabetes or insulin resistance actually here. This is not only diabetes, this is insulin resistance. And then 2 to 4 years before, 5 to 7 years, 7 to 10 years before. We see 7 years before people develop insulin resistance, the glycans are already changing in a direction of a disease phenotype. And these changes also are predictive of both prevalent and incident complications once you have diabetes already. So these are the total plasma glycans. We have no idea what is the functional significance of individual changes. So we are trying to look into individual proteins to look what could be the mechanistic role of these changes. And this is, for example, we developed a method for high throughput alpha-1 acid glycoprotein glycosylation at the glycopeptide level. So we can see the changes at the individual protein, individual glycosylation site. They're highly predictive of future diabetes, but still we have no ideas what are they doing because we don't even know what alpha-1 glycoprotein is doing, although it's one of the major acute phase proteins. There are a couple of posters if you want to learn more. The most interesting here is you can actually fix it. So by doing interventions which are known to be beneficial to prevent diabetes, the glycans change in a direction which is opposed to the disease risk. So you're kind of decreasing your disease risk by doing healthy things. And this is the part I will focus in the last part of my presentation. And this is, can we revert the glycans? Can we make a decision? can we just decide to do something and then change our glycans? So there is a lot of anecdotal data. For example, Tim Spector is a famous genetic epidemiologist from King's College. We are tracking him for nearly a decade now. And then at one point, at the peak of the pandemic, he was really bad. He decided to improve. And he improved for 11 glycan years in 6 months. We even recorded an Instagram Live to show what he did. Of course, for him, this is highly relevant. It does not have to be relevant to any of us because we have to do proper studies to see what actually works. And so far, we have shown that the weight loss works in majority of people. For example, in extreme obesity, bariatric surgery can really be beneficial. We had a patient who lost 37 glycan years in 6 months. But it doesn't have to be so drastic. When we were tracking the 2000 twins in 3 time points, we saw that twins who were gaining weight were aging faster than twins who were losing weight. So just a small change in a body mass can have a significant effect on the glycan composition. Also, we know that the caloric restriction works more or less in everybody. So we had a large study, 1,000 people on a low-calorie diet for 2 months in 8 centers in Europe. More or less, they all improved. But caloric restriction is brutal. Nobody wants to do it, especially not for a long time. So we analyzed 5 different diets as a maintenance diet. And we learned there is no magic diet. So each of the diets, even the high glycemic index diet, was good for some people and bad for some other people. So this is highly individual. We are all different. There is no standard human. The same goes for exercise. There is no standard human. What we have learned actually is that majority of middle-aged people, when they start exercising, they overtrain. So exercise has to be carefully tuned for an individual. Hormones are extremely important. Both testosterone and estrogen are strong effectors on IgG glycosylation, but testosterone works through estrogen. So in people who are optimizing the hormones, if they block the aromatase to have the testosterone high, they're actually doing the damage. This was a very interesting placebo-controlled randomized trial published in several publications the last few years. And what we learned here is that IgG glycom actually integrates information about the concentration of estrogen in a couple of weeks, which is very informative in the early stages of perimenopause when the estrogen goes up and down like crazy. So you cannot measure the hormone level because it tells you nothing, but you have a natural integrator of that level. This is IgG glycan composition, which is very similar actually to how we use HbA1c to monitor glucose overload over time. Microbiome extremely important. If you change your microbiome, it will change your glycom, but it goes the other way. Glycom affects also the microbiome. We have a couple of publications in that field. And the most important message, actually, the key message of my talk today is that disease is an endpoint. It's usually beyond the point of no return, but it's a process of over a decade. And if we wait for people to come for help, it's too late. We cannot actually help. We can only alleviate the symptoms. But if we find people when only biomarkers are affected, then an early intervention can maybe even prevent or at least postpone the disease. And the best analogy I can make is our cars. We know that we have to regularly maintain our car. Imagine if we would change tires only after they explode on a highway. How dangerous would driving be? And this is what we do to our body. We ask for help only when something stops to function. And there's a whole longevity industry which is trying to help people avoid consequences, avoid age-related diseases. Glycan age is one of the tests which we believe can actually help people because we know what is healthy, but we don't do it because the information about the beneficial effects comes too late. And with the test, it can come much sooner, and we see it on real people, that people actually modify their lifestyle once they get this information. At the very end, I have to acknowledge that our research is very generously funded with the multiple grants. And at the very end, we'll have a human glycom project meeting in November this year in Split. We are very interested to collaborate with people who have not worked with glycans before. The conference is by invitation only, so email me if you want to come. We'll have a very interesting brainstorming there. and I thank you all for your attention. And I'm sorry I'm late, I can't see the timer. There's no timer here.
Thank you so much for this fascinating talk.
Thank you. We have time? Okay. Tamas.
Thank you. This is really fascinating and eye-opening, really. I have a question about the relationship between glycosylation and inflammation, which is probably the area that I could perceive the best from this panoply of changes that you presented. So are these changes causes or consequences of inflammation? And related question is that does the rate of protein synthesis alter the composition of the glycosylation of that protein?
So cause of consequence, both. In some aspects it's the cause, in some aspects it's the consequence. A rate of synthesis changes glycosylation in vitro. So you have an expression system, it does. But for example, for IgG, it's very tightly regulated. And big part of IgG glycosylation is actually happening after the protein has been released. So during either recycling or somewhere in circulation, especially in inflammation, IgG gets reglycosylated in a process which still we do not really understand. So it's entire level of complexity, which I personally believe enabled us to become multicellular, which we are failing to appreciate because we simply do not look. And I think if we look, we will learn much more, especially because the large part of individual variability is glycan-based. Even blood groups are chemically glycans.